Hey everybody. Today we're going to do the macromolecules lecture in two parts. So for this first one, I will talk about carbs and lipids. First, I'll go over everything you need to know between the two lectures, both about carbs and lipids, and then also proteins and nucleic acids. So after these presentations, you should understand the four key macromolecules. So you should know everything about carbohydrates, lipids, proteins, and nucleic acids, how we combine and take apart polymers, the four structural levels of proteins, and what the consequences of any changes to those four levels are, how proteins configure into their final shape, and what denaturing is, and then how directionality influences the way that macromolecules work. And so in nucleic acids, that's going to be 5' and 3' ends, and proteins, amino and carboxylins, what, the different, 1,4 glycosidic linkages are, things like that. So first thing we're going to talk about are carbohydrates. And when you think carbohydrates, you're going to think energy. And so carbohydrates are composed of carbons, hydrogens, and oxygens. And this is going to be something that you're really going to want to commit to memory, is what these macromolecules are made of and what elements make up their structure. A lot of AP questions are going to target on, do you know the differences of what these different macromolecules are made of? And so an easy way to remember this is if you kind of break down the name. And so carbo represents the carbon, the represents hydrogen, and then 8 is that oxygen component. And so the function of carbohydrates is energy and energy storage. They are, energy is, they're the raw material for that energy. And then they're also going to provide structure in some organisms. And so the monomer of a carbohydrate is a sugar. So remember back from your introductory biology class, a monomer is, the single unit that makes up a polymer. So a monomer is one, poly is many, and so a monomer is a sugar, and a polymer is going to be a carbohydrate or a polysaccharide or something like that. So examples would be sugars, starches, and cellulose. So sugars, most of them are going to end in OSE. And so if you ever are, working with a molecule that ends in OSE, you can be pretty sure that it is a carbohydrate. Obviously, sometimes if we look at, this glyceraldehyde down here, that's not always going to be the case, but a lot of sugars end in OSE. And so they are also named a lot of times for the number of carbons that they have, and that is what goes into their naming. So sugar structure. Sugars like to form rings in solution. And so, you will see them written oftentimes in this linear form, but know that they like to circle up, and that's usually the form that these sugars are going to take in solution. So if we were going to number carbons, okay, if you have this carbon ring here, you're going to start from the carbon that's immediately to the right of an oxygen. So this would be your first carbon, second, third, fourth, fifth, and sixth. And you're really not going to need to know, a ton about the numbering of carbon for AP Biology's purposes. If you ever take organic chemistry, the numbering of carbons is going to become a lot more important when it comes to naming organic molecules and things like that. But we will be interested in it mainly for the type of bonds that these carbon chains are going to make. And so that, that's really why. It's just so you know where the numbers are coming from. And just remember that a lot of energy is stored in the carbon-carbon bonds. And so anytime you have a carbon bonded with another carbon, we know that that is a nonpolar covalent bond, and so it's going to store a lot of energy. So let's talk about some of the different sugars that we have. So monosaccharides, those are simple sugars. They only are, one carbon ring. Glucose is going to be the most common monosaccharide sugar. Then we have sorry about that. Then we have Then we have disaccharides, which are made up of 2 monomers or 2 carbon rings. And so sucrose is a really common disaccharide. And then polysaccharides are going to be long stretches of sugars all put together. They're large polymers, and the most common one that you will be familiar with is starches. So how do we make a sugar? We make a sugar by taking away water. So anytime you have 2 carbon rings, so here we have glucose and glucose, we're going to take an OH group away from one, we're going to take a hydrogen away from the other, and then we're going to form something called a glycosidic linkage. And so when you put 2 molecules of glucose together, you get maltose, a disaccharide, and this is that glycosidic linkage I was talking about. A lot of times they'll call it a 1,4 glycosidic linkage. Sometimes they make a 1,6 glycosidic linkage. But all that's really talking about are the carbons that are involved here in making that glycosidic linkage, and that's why it gets that name. And maltose is a common sugar that's found in beer. Polysaccharides are polymers of sugars, and so it costs a little bit of energy to build these polysaccharides, but we can reverse that and release energy in the process. And so the whole point of polysaccharides is just to store energy. And so in plants, that's going to come in the form of starches. In animals, it's going to come in the form of glycogen. And this glycogen is going to be stored in liver and muscle cells. And, when we think about a lot of that is for function, right? Our muscles are going to need a lot of energy for movement. So that's why we would want glycogen stored there. We'd want a lot of carbohydrates stored there so that we can use that energy to move. And so polysaccharides are also going to provide structure in some organisms, like plants. And so in plants, cellulose is what helps build the cell walls in plants and provides the structure for plant cells. And then chitin is something that you're going to see in arthropods and fungi like mushrooms. And again, those just provide structure for the cells in those organisms. So I'm going to talk briefly about linear and branched polysaccharides. And so linear polysaccharides are things like amylose. They are going to be completely made up of these 1 to 4 glycosidic linkages. They will kind of form a helix shape when they're stored in cells. And branched polysaccharides are a little bit different. They're going to contain both those 1,4 glycosidic linkages and then also something called a 1,6 glycosidic linkage, and that's where you see this branching happening, like in those places there. And so the difference between the two is just the way that the energy is stored. And so the branching, what that really does is it just allows these long polysaccharide chains to be stored more easily. Like here we see it in animal tissue and these like glycogen granules. And so that's just areas where a bunch of glycogen is stored. In plants, amylopectin is the way that it's going to store its starch. And so the difference between them is just that in plants, you're going to have a slow release of this energy. If we think about plants and just kind of, the difference between plants and animals, plants are making their own energy. They're making their own glucose through photosynthesis. And so, they're going to store up these polysaccharides, but they don't need to use that energy as often as an animal cell will. And so glycogen is really made for that fast release so that animal cells can have it readily available for energy use. And so when we talk again, like also about polysaccharides, the molecular structure determines the function. And so if you look at alpha glucose and beta glucose, they are almost identical molecules except for where this OH group is. And that makes all the difference. So these are isomers. They are, mirror images of each other. And so this structure is going to determine how, these glucose molecules are used. And so glucose is used in starch, is used in glycogen, in amylose, things like that. It's used for storage of energy and to be used as energy. Beta glucose is used as in the polysaccharide cellulose, and so its function is for structure in these cells. And so just like they are different in that they're isomers, they're mirror images of each other. The structure again is going to change the function of these polysaccharides. And so starches are really easy to digest. And the reason for this is because it is easy for an enzyme like amylase to break this bond and break off these glucose molecules to be used in cellular respiration. And so like things like breads and those types of foods that we eat, our body has the enzymes that are going to easily digest those starches. Cellulose, on the other hand, is really hard to digest. And it's because our enzymes that we have in our body are not equipped to handle this different bond that we have here. And so, when it comes to how we get sugars in our diet or what we get sugar from in our diet, that's going to really change the types of foods that are going to be easiest for our body to digest. So why is cellulose, why does it work for some organisms and not others? So cellulose is the most abundant compound on Earth, and that's because it's in all plant cells. It's used for that structure in plant cells, and so we're going to see it a lot. Herbivores have evolved a mechanism to digest cellulose. Most carnivores, though, we have not. And so that's why, we have to supplement our diet with meat and fruits and things like that so that we can get our energy and nutrients. And so we still eat plants, right? Like we still eat lettuce and we still eat, green leafy plants. But that cellulose that we're eating is undigestible and rough. And so when you think about that, this undigestible roughage, that is going to be fiber. Okay, so when people talk about having a high fiber diet, it's really that stuff that you can't digest that is kind of just lining your intestines to make everything come out a little smoother. And so, that's why that's happening, is because of cellulose and our inability for our enzymes to digest those bonds. And so what types of organisms can digest cellulose? So cows can. They can digest cellulose well, and so they can completely live off a plant diet. They don't need to get sugar from anywhere else. However, a gorilla cannot digest a cellulose wall. And so they're going to be, their diet is going to be more similar to ours, where like if you were a vegetarian, you're going to have to get your sugar from things like fruits and just in other methods. So how are cows able to digest cellulose? It's because they have a symbiotic relationship with this bacteria that lives in their digestive system. And so what happens is they have ruminants or bacteria that live in their gut, and the bacteria help digest that cellulose that's in their system, giving energy to the cow and also giving the bacteria a nice happy place to live. So a symbiotic relationship, they both benefit from it, and that's how a lot of herbivores are able to eat these plant-rich diets. All right, let's move on to lipids. And lipids are our long-term energy storage. So, this is, if we have to go into energy reserves, we're going to get that from lipids. And so lipids are composed of carbon, hydrogen, and oxygen, and sometimes phosphorus, okay? And we're going to talk about exactly when they're composed of phosphorus, not all the time, but mainly they're going to be composed of carbon, hydrogen, and oxygen. And so they're made up of these long hydrocarbon chains, which we remember are just really long repeats of carbons and hydrogens. And when we think of lipids, we can kind of break them into three main groups: fats, phospholipids, and steroids. And when you think of lipids, we're going to think of them as big fat molecules, okay? They don't form polymers. So they're just going to be one big monomer and never make a continuing chain or anything like that. So the structure of fats is a glycerol. So that's just a 3-carbon alcohol. Right here is that glycerol component, and then that fatty acid. And remember, the fatty acid is just a really long carbon chain, okay? And then it has this carboxyl group on the end of it. And so, again, to make our fatty acids, we're going to do a dehydration synthesis reaction, okay? So again, we're going to take an OH group from that carboxylic group. We're going to take a hydrogen from the glycerol. We're going to have water leave this reaction and then put one of those fatty acids onto the glycerol chain. And that's going to happen 3 times. And so we're going to end up getting this molecule that looks like a big letter E. And when you think of this E, think of ester linkage, okay? That's what you call this link between the carboxylic group and the glycerol. And so that's an ester linkage. When you have dehydration synthesis, that is what you make. And so fats store energy. So fats are nonpolar, okay? And they are hydrophobic, all right? They don't like water. And so the reason they function with this energy storage is because of all these carbon-carbon and carbon-hydrogen bonds, okay? They have so many of these non-polar covalent bonds. They're going to have a lot of energy in them. But it takes a little more energy on our part to use these for cellular respiration or to use them for immediate energy, and so that's why they're reserves. And when you think about, what lipids are doing, think about them cushioning our organs and insulating our body. That's kind of, the job of fats. So there's 2 main types of fats, and this is going to be really familiar to you. We have saturated fats. And so in saturated fats, all of the carbons are bonded to hydrogens and one other carbon. There's no double bonds between the carbons. And so what that's going to give you is just a straight chain of carbons. And this saturated fats, if you think of saturated fats, you're going to think animal fat, like butter. And they're solid naturally at room temp. And as you know, animal fats contain a lot of cholesterol. And so they're usually, these saturated fats are linked to cardiovascular disease and putting plaque deposits in our cardiovascular system. And so a lot of times saturated fats are thought of as kind of like bad fat for people to have. And so this is what it looks like when a saturated fat is just a really good straight letter E. And so the opposite of that are unsaturated fats, okay? And so in unsaturated fats, we are going to have these carbon-carbon double bonds. And so what that's going to do is it's going to make a kink in that hydrocarbon chain. So anytime you have that carbon-carbon double bond, you're going to get a kink. And so because of those kinks, it makes these fats liquid at room temperature. And you're usually going to see unsaturated fats coming from plants and things like that, so like your oils. And so these kinks make it hard for these letter E's to pack nicely together, okay? Those kinks that they have make it so that they can't nice and tightly pack, and so that's why they're going to be more liquid at room temp. And so a lot of times these unsaturated fats are thought as healthy fats or fats that are better for you. But beware of trans fats, okay? And so trans fats are transformative fats. And so pretty much what happens in transformative fats is they take, they market them as, healthy fats because they're not made with animal fat. They're made with oils and things things of that nature. And so what they do is they modify these carbon-carbon bonds and they make them straight again so that they can be solid fat. They can have the taste of solid fat. But because molecularly we've made this change, our bodies are not always equipped to handle transitive fats. And so an example of that are these WOW chips. They were something that kind of came out in the 90s, and they were marketed as like this chip that, it has all the taste of, regular animal fat, but, it hardly has any fat. But really what they were doing was just, modifying the fats so that they seemed healthier, but when people actually consumed the chips, they didn't have the proper enzymes to break down the fats, and it just ended up making them sick. So that's what, and we're finding more and more out about transitive fats and how they're not good for us, but that is one reason why trans fats are not always so great. Okay, let's talk about phospholipids. So a phospholipid is a glycerol, so just kind of like when we were talking about fatty acids. It has a glycerol group, it has a fatty acid on it, but then it has this phosphate group on it. And this phosphate group is what is going to give these things a split personality, because this phosphate group is negatively charged. And so what that's going to do is give this molecule kind of a bipolar trait. So on one side, it's going to be hydrophobic, and it's going to fear water. And on the other side, it's going to be hydrophilic and love water, okay? And so that's what we mean when we say like a split personality. And so what that's going to do is it's going to make the interaction of a phospholipid with water very complex, but it's very important. And so why is it important? It's because phospholipids are what make up our cellular membranes. And so it gives us that water barrier. So inside the cell membrane, you're not going to have water, and on the outside, you will have water. And that's because these phospholipid heads will orient themselves towards water, and the fatty acid tails will invert themselves so that you have a space without water and a space with water. Steroids are the last type of fat we need to talk about. And so they are 4 fused rings with a different functional group on the end, and that functional group is really what determines their function. Examples of steroids are things like cholesterol and sex hormones. And so cholesterol is actually kind of another bipolar type of molecule, usually we think of it as something bad. But when we talk more about the cell membrane, next unit in cells, we'll kind of learn about cholesterol's kind of the good things that it does in terms of our cellular structure. And so it's not always a bad molecule. Okay, so that is all for our lipids and carbohydrates. And the next video, we'll talk about